Your Module 6 Stat Lab assignment is the One Way ANOVA Judging Art Lab. Before you complete this lab, make sure to read Chapter 12 in your textbook on the logic of ANOVA. At the opening of the lab, you'll get some background information on the One Way ANOVA. There's going to be a total of 33 trials in this lab. What you'll be doing is viewing abstract art images and using a slider to rate the happiness level of that image under one of three conditions, and you'll read about them here. The first condition is a neutral condition where you just normally use the slider to make your ratings. In the second condition, you're asked to take a pen or pencil and bite on it with your teeth while you make your ratings. That is simulating a smile. And the third condition is holding a pen or pencil with your lips, which will simulate a frown. So you'll have 11 trials at a neutral condition, 11 trials in a smile condition, and 11 trials in a frown condition. Once you've completed all of your trials, you'll get your standard output in terms of questions. Make sure to pay particular attention to the questions section in terms of independent and dependent variables, as well as predicted hypotheses. Stat Lab also has information on how the ANOVA is calculated, and if you want to do the calculations in Stat Lab, you can, but as in previous assignments, it's recommended that you import your data into Excel. You'll see three columns: the trial number, the condition. They'll all be sorted by condition. Now yours may look different than this because I think the conditions are randomly assigned. So you may end up doing the frown condition first and then neutral and then smile or whatever. But in the end, you'll have all three conditions sorted and then your happiness ratings for those conditions. You'll want to take this data, of course, copy it and paste it into Excel as you've done in your previous assignments. I went ahead and did some of my work ahead of time for this video. And so when you first start, you'll paste in your standard output from Stat Lab. In order to run the One Way ANOVA in Excel, you have to do an additional step. If you were to try and run the ANOVA as it's set up from Stat Lab, you'll get an error message in Excel. So we have to do one additional step before we can run our analyses. What you'll want to do is in three new columns, create each of your three conditions and label them to help you keep them, help you track your various conditions. So I recommend no pen neutral, lips, which is frown, and teeth, which is smile. Under each of these labels, you'll then want to paste in your happiness ratings for that individual condition. So for example, for no pen, for me, I would copy and paste my happiness ratings, and you'll want to do that for all three conditions. Make sure you set this up properly or your ANOVA will give you an error message. The next step, of course, is some descriptive statistics. You'll want to calculate the mean and standard deviation for each of your groups. You'll need that for your paper. Before we run the actual One Way ANOVA, let me show you the paper and discuss it a little bit. The first section, of course, you're familiar with by now, the introduction, discussing the context of the study, articulating your independent and dependent variables, as well as the predicted outcomes. And you'll get that information from the questions section of Stat Lab. The second step, visual representations, asks you to create a summary table of your ANOVA. So in order to do that, we actually have to run the ANOVA at this point to write up the second step. I've already done this in advance, but I'll show you how to do it in a separate workbook. To run the One Way ANOVA, make sure again that you have your columns set up properly first. Go to your Data Analysis button, and the very first option should be ANOVA Single Factor. Select that, press OK. Let me clear this out. Because I've provided labels for my columns, which I strongly recommend to help you keep them straight. Select labels in first row, and then go ahead and select your data. And I'll go ahead and create this in a new workbook for this video. Make sure also you're set at columns and press OK. And this is the ANOVA results. The first column will be squished, so you want to pull that out a little bit. You see here we have a summary statistics for the 3 conditions and my average happiness ratings for each condition. So what the One Way ANOVA is going to do is tell you whether or not there is a significant difference somewhere between these means. In order to determine that, you go down to your ANOVA table and you look at your probability value. You learned, of course, when you see an E, this is scientific notation for a very small number. You can actually check this by going into the number tab and then expanding out your decimal places. Recall that any probability value less than 0.05 is significant. Reject the null hypothesis. So what the ANOVA is telling us is that there is indeed a significant difference somewhere in these means. It doesn't tell us which means are different from one another, which we'll cover in a little bit. But this is basically your summary table for ANOVA. It's going to be fairly straightforward in terms of creating your visual representation. Let me show you how I set that up. You'll want to check with your instructor if they have different preferences. But the way I set up my summary table is as follows. Mostly just translating these values straight over into the table. You have the source, which is your various sources of variance, sum of squares, degrees of freedom, the mean square, the actual F value, and then the probability value. And because this is such a small number, generally in APA format, we just go out to the 3rd decimal place. But again, what this summary table shows is that the overall analysis of variance is significant, at least in my data set. So you may be wondering, moving forward, why we don't just run multiple t-tests. We've already learned about t-tests. Why do we need ANOVA? You can actually run multiple t-tests. You could run a t-test between this group and this group, this group and this group, this group and this group, for a total of 3 comparisons. The problem with that, and the reason that we don't teach running multiple t-test comparisons, is what's referred to as inflated type 1 error rate. A type 1 error is when you reject a null hypothesis when you shouldn't reject it, and that's a problem of interpretation. Statisticians have discovered that the more comparisons that you make with t-tests, the more likely you are to make a type 1 error. So with your standard t-test, which has 1 comparison, your alpha level is 0.05. However, the more comparisons that you make, the more likely you are to actually commit a type 1 error, which is an error of interpretation. Basically, you're saying that something is significant when actually it's not significant. So we never run multiple t-test comparisons with the same data set. And that's the reason why we use the One Way ANOVA, is it protects us from inflated type 1 error rate. In order to do your comparisons, we're going to use what's called a Bonferroni correction. Here you see the 3 groups with my happiness ratings, and the overall ANOVA told me that there is a significant difference in means somewhere, but we're not sure where. We have comparison 1, which is A by B, comparison 2, which is A by C, and comparison 3, which is B by C. So we have a total of 3 t-tests that we're going to run. But to protect us from inflated type 1 error rate, we have to adjust our alpha level. The Bonferroni correction is a very simple calculation. It's simply 0.05, which is your standard alpha level, divided by the total number of comparisons. And we have 3 comparisons we need to do. So what we're going to do is we're actually going to adjust our alpha level to 0.0167. That's 0.05 divided by 3. When we make that adjustment and run our t-tests, that will then protect us from inflated type 1 error rate. So the next step in the process is to actually run your t-tests for these various comparisons. The caveat here is that when you're working on your paper, if you run your one-way ANOVA and the probability value is not significant, you stop. You're not actually justified in running post hoc tests unless your overall ANOVA is significant. So again, when you're writing your paper, you run your one-way ANOVA. If your p-value is greater than 0.05, you stop and do not reject the null hypothesis. However, if your p-value is less than 0.05, you have to move on to report your Bonferroni corrections, and that's stipulated in the assignment. So we'll go through the process of actually running Bonferroni corrections. I've already done these in advance, but I'll show you how it's done as a refresher. So our first comparison is no pen to lips or neutral to frown. We want to know if these two mean scores are significantly different. So we run a standard t-test, go under data analysis, and select t-test to sample assuming equal variances. Press OK. Let me clear this out. Again, since I have my labels in place, I'm going to select labels, and I recommend using labels for this assignment because there are 3 conditions, and it's easy to forget which is which. Variable 1 will be our neutral condition. Variable 2 will be our lips condition, our frown condition. And we'll go ahead and open this in a new workbook so you can see. Remember, our new alpha level is 0.0167. They're all squished. These columns are all squished, of course, so you pull them out. Now this is our A by B comparison. We look as we did in the past for the t-test, for the two-tailed version of the t-test, and we see here that the probability is 0.03. So if we were running a standard t-test, this probability is less than 0.05. So we would conclude that there is a significant difference. However, remember that the Bonferroni correction is 0.167, which is lower. In this case, the interpretation is that they are not significantly different because the value of 0.03 is greater than 0.0167. So when we run the t-test with a Bonferroni correction, we conclude, at least in my data set, of course, that these 2 conditions are not significantly different. I actually went ahead and ran all of my pairwise comparisons ahead of time. So group A to group B, this would not be rejected. Group A to group C, this is less than the Bonferroni correction. So we would conclude, given my data set, that there is a significant difference between the neutral condition and the smile condition. And you can see that my ratings when I was simulating a smile are significantly greater than my ratings when I was in a neutral condition. And then finally, the final comparison is the frown to smile. And again, this is a very low value. We can check that again here under number and just bring out the decimals. You can see here that there is also a significant difference in my data set between the frown condition and the smile condition. And those are all results that you would want to type up in terms of your analysis. So again, for me, with the Bonferroni correction, the neutral to frown condition was not significantly different, but the other two conditions, the other two comparisons are significantly different. So when you write up your results, if you get a significant ANOVA, you then have to include your post hoc tests where you report your t-tests with a Bonferroni correction. After you've written up all of your results, you then interpret those against the null hypothesis. If you have any questions, please reach out to your instructor. Thanks.